The lecture you are about to see is part of our annual Alan Edwards Psychology Lecture Series. Professor Edwards was affiliated with the Department of Psychology for half a century until his death in 1994. He was an outstanding teacher, researcher, and writer who introduced new statistical techniques that are credited with changing the way modern psychological research is conducted. Alan also permanently enhanced the intellectual climate at UW Psychology by endowing the Alan Edwards Lectureship, which since 1999 has brought in an impressive list of renowned psychologists to the UW campus to interact with faculty and students. The lecture you're about to watch is one of a pair given back-to-back that matched a UW psychology professor with a visiting researcher to talk about a topic of great public and scientific interest.
It is my pleasure to introduce our second speaker this evening, Dr. Richard Fay. You'll notice that Dr. Fay will be seated for this evening's lecture. He can tell you more about it, but was in a skiing accident several weeks ago on the slopes of Vermont, but bravely made the trip out here to Seattle for this evening's lecture. Dr. Fay is presently an adjunct scientist and Whitman investigator at the Marine Biological Laboratory at Woods Hole and an emeritus professor at Loyola University in Chicago. Dr. Fay is a world-renowned expert on fish hearing and has major research interests in the neural mechanisms of perception, sensory neuroscience, and the evolution of hearing. He received his BA in psychology from Bowdoin College, a master's in experimental psychology from Connecticut College, and a PhD from Princeton in 1970. Following his PhD, he was a postdoc in the lab of George von Bekesi, a Nobel Prize winner at the University of Hawaii. And then Dr. Fay joined the psychology faculty at Loyola in Chicago as an associate professor in 1975, where he spent the rest of his career. Dr. Fay has published over 150 publications in peer-reviewed journals and book chapters, including in Science and Nature, and has been funded continuously by the NIH and NSF during his career. He's been a series editor for the Springer-Verlag Handbook of Auditory Research since 1991, with over 43 volumes to date. And he's currently an associate editor of the Journal of the Acoustical Society of America. Please join me in welcoming Dr. Richard Fay.
Well, thank you very much for that introduction. And I want to thank you all for your attendance here. You're really hanging in there. This is 2 hours of lectures about fish is a little unusual. But I want to thank the Alan Edwards Lecture Series for inviting me, and Joe Cisneros from the Psychology Department and the Department of Psychology for inviting me to speak here. It's a real pleasure to speak to you today on this topic, the sense of hearing in the goldfish. I think I've had a fairly unusual career in science, in that I decided as a graduate student what I wanted to work on and what I wanted to know, and I've been lucky enough to be able to do that throughout almost a 50-year career, and to study this topic, the sense of hearing in goldfish, for about 45 years. Some would say this is compulsive on my part, and I think that's true. It is compulsive on my part. But I feel really lucky that I was able to do this. Now, my interest in fishes began as a kid because I like to catch fish and eat them and, cook them for breakfast. When I did the first experiment that I ever did on fish in 1964, I chose goldfish to work on, and for all the wrong reasons. It was because you could buy goldfish at Woolworth's for 50 cents anywhere in the country. And so that's what I did. I went down to Woolworth's and I bought some goldfish and started some experiments. I continued working on goldfish, though, because they offered an interesting opportunity. And the opportunity was to study a fish that heard well, but at the same time didn't communicate with sound, like the midshipman fish, for example, communicates with sound. As far as anybody knows, the goldfish doesn't make a sound and doesn't communicate acoustically in any way. So this led me to have to come face to face with the question of what in the world does the goldfish listen to right away? Because if it made sounds, you know, I could just dismiss the question, say, well, of course they listen to they listen to their vocalizations. There's no such explanation possible for the goldfish. So I had to think about this question, you know, on the very first day. Had to really start to think carefully, what are they listening to? Because there's no simple answer to it. So I started my research with behavioral studies, because after all, I was trained as a psychologist, and I was in the psychology department, and psychology is the study of behavior. So that's where we all started. And I started, though, with two kind of classical classical methods in psychology at the time. One was the very old method of classical or Pavlovian conditioning, 1910, and which I'll describe as I go along. And the second method of psychology that I used was what's called psychophysics, and it's the quantitative study of sensory behavior, and it's what 19th century psychologists did. You know, that's about all they did. You know, psychophysics, and it came into the 20th century a little bit, and there's still a very small number of psychologists who continue to use psychophysical methods, but a small number. My interest in psychophysics came from my training in psychology, where my interest in hearing also came from my training in psychology, because that was a topic of interest, and it was the only discipline for a long time. It's not true today, but for a long time it was the only discipline, psychology, where you could study hearing in its entirety. So I had this interest in fish, and I had this interest in psychophysics and hearing, and when I started to delve into the neurobiology or the physiology of hearing, which I did, the questions that I asked, however, always arose not necessarily from the physiology that I was looking at and studying, but they arose from the behavior. You know, so every question that I asked was sort of motivated by explanations for the behavior of the fish. And my questions at the end were often asking whether goldfish heard or had a sense of hearing anything like human beings. And so that's what I'm going to talk about today. Do goldfish hear or have a sense of hearing anything like human beings? The very first experiment that I did was a rather boring experiment, a conventional experiment. It was the determination of the hearing range and sensitivity of the goldfish. And it was to get one of these curves that's called an audiogram. And there's the audiograms for humans and all kinds of animals in the literature, and there's audiograms for fish. And so the first step in hearing, to study hearing in fish is to determine the audiogram. And this slide illustrates one of the nightmares of audiology in animals. And that is that here are, let's see, 6 different audiograms determined all about sometime in the 60s for a goldfish, the same goldfish. And they differ just wildly. You know, there's 60 decibels or a factor of 1,000 difference between a threshold or a measurement that one person would get and a measurement that another person would get. And so while you do what you can to assure that you're designing a good experiment and manipulating your variables properly, you just don't know. And so the question here is which one of these is the valid audiogram for the goldfish? Which one describes the goldfish best? And I don't know. You know, and there's no way to know. We'll never know, really. And it's sort of humbling to realize that. On the other hand, all these audiograms have something vaguely similar. They have something in common. They all have a best sensitivity to frequencies around 400 to 1,000 hertz, which is low for mammals and even low for human beings. They all have a basically U-shaped audiogram. And they all have a loss of sensitivity at higher frequencies, above 2,000 hertz or so, and pretty good sensitivity. Most of the audiograms show pretty good sensitivity at low frequencies. And so the audiogram is sort of the necessary first step if you're starting a research program on the sense of hearing in the goldfish. The very first thing you have to do is determine what the audiogram is, so you know what sounds to present to the animals and how audible those sounds will be once you present them. And so I went on to study many other aspects of the sense of hearing of the goldfish beyond this hearing sensitivity. And I'm going to tell you about several of them today. But before I do that, I want to talk about the method that we developed for studying hearing in the goldfish. And as I mentioned before, that method involves classical conditioning or Pavlovian conditioning. And I want to tell you about that now. This is the kind of a diagram, and it illustrates some of the features of this classical conditioning. So a goldfish, a pretty good-sized goldfish, 6 inches long, is gently restrained in a cloth bag and put in a little body of water with a loudspeaker underneath that's producing some sound. And the only thing the fish can do is sit there and respirate. All it can do is gulp, and bring water over the gills. But they'll do that, they'll sit there for hours and without any ill effect, just respirating. And so if you present a shock across the fish's body from head to tail, indicated by shock electrodes at the top there, there's an unconditioned response to this shock. It's a mild shock. It's not. It can't be too strong, or it defeats the whole experiment. It has to be mild. And the response to the shock is the animal closes its mouth and just stops respirating for a couple of seconds or so, and then slowly begins again. And so that's the unconditioned stimulus, and that's the reflex. Nobody knows why or how this reflex happens, but it does. So if you precede the shock with the sound repeatedly, and the sound that precedes the shock would be called the conditioned stimulus, and that's down there on the third line down. In a sound detection experiment, the very first line labeled sound detection indicates that during the conditioned stimulus a tone comes on, beep, 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 and that's the signal that doesn't produce any kind of response to begin with, but when paired with a shock, after a few trials, maybe 10, maybe 20 times when this is repeated to the goldfish, the tone takes on some of the characteristics of the shock. It tends to produce a suppression of respiration, just like the shock does. And and that's your signal, that's your indication that the animal has heard something. If it suppresses its respiration to the tone, like this waveform up there on the screen, that's a conditioned animal. That suppression of respiration during the tone is an indication of hearing. With a method like this, you can investigate all kinds of things besides sound detection. So the very first line up there is sound detection. A tone comes on out of silence, and that's the conditioned stimulus. And the second line, it's indicating that the frequency of the tone, the pitch of the tone, beep, 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 before the CS, just changes. The CS is a change in the frequency. So the frequency may go up up or down during the conditioned stimulus. Or it could be a level change or a loudness change. The tone all of a sudden becomes louder during the conditioned stimulus. Or it could be a signal added to a masker, or a temporal pattern change, click rate, or click repetition rate, or anything that you can imagine can become a conditioned stimulus in this, and you can study the fish's response to it in the same way. And so with this kind of method in mind, the very first thing I wanted to do was to study frequency discrimination. And frequency discrimination, or pitch discrimination, is just a discrimination between two tones that are identical except for the pitch. And so there's a history to this question. Because Carl von Frisch, in 1929, was very interested in this question of pitch discrimination in fish. And the reason he was interested is that the fish, he recognized that fish do not have a cochlea like the mammalian inner ear. They do not have a basilar membrane where frequency is analyzed according to place of excitation. And so the question arose, okay, if they don't have an ear like this that can make sense out of changes in frequency in the same way, how can they make sense out of any change in frequency? Can they do it? And if so, how do they do it? And so this was the question at the early part of the 20th century that Carl von Frisch was quite interested in. And so we looked at frequency discrimination. And so this is kind of gives you a summary of the way I tended to look at things. And the way I tended to look at things is the goldfish, which is the red curve, is in the context of all the other vertebrate animals that had ever been tested in this same way. You know, let's look at it compared to everybody else, including humans. And I don't think you can read that slide, or the labels on that slide, but the human thresholds for frequency discrimination are the three bottommost curves. Humans are very sensitive to changes in frequency, and they have very low thresholds. And all the other curves are cats and monkeys and bush babies and pigeons and tree shrews, guinea pigs, white rats, all the other animals that have been tested up to that point. The red, as I said, the red line is the goldfish, and so you look at this result, and it says, well, you know, the goldfish are not as sensitive as humans. That's obvious to changes in frequency, but they're no less sensitive than some other vertebrate animals, including some mammals, and they have a function that is reminiscent in slope and form to all those functions on the screen. In fact, you could say, if you squint a little bit and look at that, you say all those functions are kind of similar. And the goldfish fits right in with them. It's similar. And so the very first question that I had when I looked at these frequency discrimination of the goldfish is I asked this question: is this kind of parallelism of their functionality of in frequency discrimination or pitch discrimination? Is this just a coincidence? Because I didn't have any basis for knowing or predicting whether it was just a coincidence or not. I said, boy, this is interesting. Gee, goldfish are just like every other animal in this. I wonder what else could be true about goldfish. So the next topic I looked at was level discrimination or intensity discrimination. And that's a discrimination between two tones that are identical, except one tone is louder than the other. And so we asked, how much louder does it have to be? And that's the intensity discrimination threshold. And this was of classical concern in human hearing since the time of Weber at the beginning of the 19th century. This is one of the questions that was most interesting to people studying hearing, and most important was this one of intensity discrimination. What is the threshold for intensity discrimination? How sensitive are we to changes in intensity? And more recently, it's been studied in terms of the effect of duration, signal duration, of the duration of a signal on the sensitivity to intensity change. So in general, as a signal becomes longer in duration, it becomes easier to discriminate from another signal of a slightly different intensity. What about goldfish? How did goldfish stack up in these terms? There's the human function and the goldfish function. And the human function shows that humans are much more sensitive. They are really, truly very sensitive to intensity change. But that as the duration of the signal gets longer and longer, the threshold or the sensitivity of the human gets better and better. But the exact same thing is true of the goldfish, even though it's less sensitive. As the signal gets longer in duration, its sensitivity increases according to a function of about the same slope as occurs for humans. So is this another coincidence? Or was the goldfish trying to tell me something? Was I, learning something? I guess one of the very next things that interested me was the question of the effects of masking noise or noise in the background on the ability to detect signals. And masking has been a very interesting topic in human auditory psychophysics for probably 100 years. And it's revealed a lot about how the human auditory system works. And it's, so everybody understands masking. When you're on the phone and there's a lot of noise in the background, you can't hear what the people are saying on the phone. Your sensitivity to signals just deteriorates as the noise level increases. And the quantitative study of masking has been related to the way that the mammal ear and the human ear work as a frequency analyzer on the basilar membrane. How frequencies are sorted out on the length of the basilar membrane, and how these facts lead to the phenomena of masking that we can study. And so the question was, well, fish don't have this basilar membrane, they don't have a cochlea. So what about masking in fish? Do they have the same, is it the same phenomenon or is it different? And again, we have this graph that shows the goldfish in red with respect to all the other animals that have been studied up to that time, including a porpoise and a seal and a cat and a rat and a chinchilla and, you name it, just a few other species that have been studied this way. But you come to the same kind of conclusion here. You squint your eyes a little bit and you say, well, all those functions have something in common. And not only that, so does the goldfish function. It has something in common with those other functions. It's about the same slope, and the masking effect increases with the frequency of the tone in the same way for all vertebrate animals, including goldfish. Now, the explanation for that result in goldfish can't possibly be the same as the explanation for the human and the mammal result. For the humans and the mammals, it's the way the cochlea works and the basal membrane works. It's a mammalian organ, and it doesn't occur in fish. But without that mammalian organ, the fish ear and brain still works according to these same principles. So what does this mean? There are many examples that I could go through to illustrate this kind of parallelism between fish and humans or other mammals, but I don't have time to do it. But what I can tell you is that in everything that I've looked at, in masking and discrimination and detection, the same kind of story comes through. There's something similar among all animals, similar to humans, and that similarity is shared by the goldfish. There's something about vertebrate animals that gives these same results. So I don't have time to tell you about all these studies, but what I wanted to do is tell you about a few other kinds of studies that I've done when all these experiments have been on detection and discrimination, things that animals can discriminate from one another and so on. And I really needed to talk about perception. What I wanted to know is not so much what can be discriminated from one to another, but what about these perception of sounds? And what do things sound like to the goldfish? That's what I wanted to know. And well, it turns out that there is a way to determine what they sound like that are called stimulus generalization methods. And every experiment that's ever been done on animals that's been done to determine what things sound like to a dog, or sound like to a cat, or sound like to a rat, or sound like to a fish, all the experiments are the same. They've all used the stimulus generalization method. Yeah. And so in a generalization experiment, here's basically how it's done. A fish or animal or person is conditioned or trained to respond to any given sound. And then it's tested for their response without shock, or without reinforcement, or without telling them whether they're right or wrong. They're tested for their response or generalization to other novel signals that they've never heard before. So the degree of response to these novel stimuli, the novel signals, will tell you if these novel sounds are perceptually similar to the sounds that the animal was conditioned on originally or not. If they respond 100% to the novel sounds, you can conclude, well, they're, as far as anybody knows, they're identical or equivalent to the conditioning sound. They're similar, they're identical. But if they don't respond at all to these novel sounds, you could say they're not perceptually similar. They're perceptually quite different, non-equivalent. And so you can do an experiment, and here's the kind of experiment that we did. And this illustrates this remarkable result from a stimulus generalization. To me, is remarkable. So in this experiment, the animal was conditioned with a conditioned stimulus originally at 166 hertz. That's the red line in the middle. And that shows a respiratory waveform and a conditioned response and an unconditioned response to the shock, its received shock. And then it's tested for all the frequencies surrounding 166 hertz, down to 79 hertz, up to 350 hertz, all without shock. And they're all presented, and in a sense, we're just asking the question, okay, you're conditioned to 166 hertz. What do you think of this? How about this one? Is this the same as 166 as far as you're concerned? Or is it different? And the magnitude of the response will tell you whether the animal thinks it's the same or responds to it if it's the same or different. And the remarkable result is no matter what frequency that you condition the animal to originally, you get an inverted V-shaped gradient of response around that frequency. And it's illustrated on the right over there. And so the conditioning stimulus, the stimulus the animal's conditioned to, evokes a response that is said to be 100%. And the response evoked by all the other tones to the higher and lower in frequency evoke less of a response. And the farther you get from the conditioning frequency, the smaller the response gets, until it just goes away completely down to zero, which means there's no response whatsoever to those tones, even though they're all very audible. They're very loud and very clearly hearable by the goldfish, but the goldfish does not respond to them. And this was a very unusual and remarkable result. And I remember when I first tried to publish this, a couple of reviewers wrote in their reviews that they just plain didn't believe what I was saying, because their learning theory had told them that this doesn't happen. It can't happen. But as far as I think it happens, and very fortunately it happens, because we could use this generalization or lack of generalization to study other features of the goldfish's sense of hearing. And so we studied we studied like 8 or 10 different aspects of the goldfish's hearing, but there's 2 experiments that I want to talk about just today. And the first one is an experiment on what I call analytic listening, or sound source segregation. And this is something that has been studied in humans using these terms. And the simplest way to study analytic listening is just to present 2 tones mixed together. So it's like a chord. The 2 tones are played, and you ask a human being, well, do you hear one entity or 2? And what's the frequency of one tone and what's the frequency of the other tone? And if the tones are separated far enough apart, listeners will tell you, oh yeah, well there's 2 there. There's one that's kind of high and one that's kind of low, and you can match the pitch of both of them, and you can determine that you're determining the pitch of both of those tones. So, but what about goldfish? What do goldfish do? How do goldfish hear those two tones when they're mixed together? Do they separate them into the entities that made them up? Or do they hear them like a chord, like a new entity, a third entity, all of its own, something unique? What do they do? And so the experiment, to make a long story short, the experiment is illustrated here, and the 2 curves at the top indicate generalization gradients around 2 tone frequencies, 166 hertz and 724 hertz. When they're presented singly, just all by themselves, pure tones, to groups of goldfish, And then the fish are tested for their generalization to adjacent frequencies. You get those two curves at the top. And that's not surprising. That's just what we saw previously. But in the bottom case, we condition the animal to respond to the two tones mixed together. Okay? And the generalization test was just single tones of various frequencies. Which tone of what frequency did they respond to the most? And what it turned out is that they responded to the tones that they had been conditioned to respond to originally, even though they were mixed together when they were first conditioned. They apparently heard out the two tones. They could analyze the two tones separately. And they could tell me that they heard 166 hertz and 724 hertz. That's what the goldfish heard. And so this is just exactly what human beings do. And so this is called sound source segregation or analytic listening. And then the second topic that I wanted to tell you about is what's called auditory scene analysis. Alfred Bregman, a psychologist, coined this term for human listeners, in 1990. He'd been studying this for quite a while, and he wrote a very influential book about it in 1990 called Auditory Scene Analysis. And it refers to the ability of human beings. His book was entirely about human beings, and it was entirely about speech and music perception. The ability of human beings to determine most of the individual sound sources that were occurring in the milieu around them, what I call the soundscape. Just whatever sound sources happen to be there in your everyday life, human beings can do this. And you all know just by introspection that you've all done it, and it's simple. And in fact, it's maybe so simple and self-evident to you that you've never even noticed that you do it. You walk outside and you hear traffic, and you hear wind in the trees, you hear birds, radios, all these things, and you assign each sound to the source that produced it. You perceptually assign each sound to a source. And it's just incredible that this happens, because the sounds, when they reach your ears, are all mixed together and cause the eardrum to vibrate just in and out along one dimension. But somehow this complex pattern of added sounds are then decomposed by the brain, the auditory system and the brain, into these individual sources that made them up. And I think one example of that is the ability to hear out the individual instruments in an orchestra. You can pay attention to the basses and pay attention to the violins and pay attention to each of the instrument types in an orchestra. And the example that Bregman used in the introduction to his book was you segregate the voice of your mother reading you a story when you're in the cradle from the squeaking of the cradle as it rocks on the floor. And if you couldn't do this as a human being, you wouldn't be able to survive. If you mixed up or conflated or confounded the sounds of the squeaking cradle and a human voice, if you did this, you wouldn't live long. And I don't think any animal would live long if they couldn't do this. So the question is, can goldfish do this? Or are all sound sources jumbled together in perception? Do the fish hear them as just a jumbling of sound sources, or does the fish able to hear them as separate entities, where it can associate the acoustic signal from one source with a specific source, and the acoustic signal from another source with a different source? And so this was the question. And so we did this experiment, and it's rather complex, and so I want to tell you just kind of what it was like. What I finally decided to do was to produce two complex sounds. The sounds were made up of clicks or transients that were repeated. And so one was a low-frequency click that I call a thud, and another one was a high-frequency click that I call a snap. And they were both repeated at different rates, and the thuds were repeated slowly, like 20 times per second. You'll hear brrr, like this. And the snaps were repeated at much higher rate, and so it sounded something like the midshipman fish with fundamental and harmonics in it, like a hum. And so these were my two sounds that I started out with. And so, so fish were conditioned to these two sounds, these thuds repeated slowly or the snaps repeated rapidly, two different groups of fish. And then they were tested in generalization to thuds or snaps, repeated at a variety of different click rates. You know, so I take the low frequency thud and repeat it at different rates, and the high frequency click, the snap, repeated at different rates. And the question was, which click rate did the goldfish respond to the most? Did they associate the click rate and the thud or the snap kind of quality together and analyze them separately? That was the question. And the answer is these are the signals that I use. These ones on the left are the thuds. The ones on the right are the snaps. The thuds are repeated slowly with a 53 millisecond period. The snaps are repeated rapidly with a 12 millisecond period. They're two different sources that were presented mixed together. So the bottom line is they were perceptually segregated from one another when they were heard as a mixture. That the fish responded, and the fish apparently knew what the click rate, what the repetition rate for the thuds were, and what the repetition rate for the snaps were independently, even though it heard them as a mixture. And so this is really good evidence for auditory scene analysis. And so humans have auditory scene analysis, and so do goldfish. And I did several experiments with this idea in mind, and got the same result both times. Now, Ted Bullock, the late Ted Bullock, who was the founder of Society for Neuroscience in the 1960s, the great neurobiologist from San Diego, wrote in 1992 that I cannot find an authority for any statement on exactly what is different with respect to the physiology of audition at its best in fishes, amphibians, mammals, apart from the upper frequency limit, apart from the fact that they hear in different frequency ranges. They can't find anything different in the physiology. And I believe this is still true, you know, like 20 years later. And I would add that we should add perception and discrimination behavior to this general statement that Bullock made. I tried and failed to determine exactly what is different between goldfish and mammals in their sense of hearing. I couldn't do it. Every time I did an experiment, they came out more similar than different. And so far I find nothing fundamentally different between the human hearing and goldfish hearing. So, my conclusions. The original question was, what do goldfish listen to? And so, in general, I think that the answer is that goldfish, perhaps all fish, and all animals, all vertebrate animals, listen to the soundscape that always surrounds them. That what they listen to is On the one hand, it's mysterious because we don't know what the soundscape consists of for any given species. We know what ours consists of, but it's hard to say what a goldfish's soundscape really consists of. But it's whatever all the sounds are that are occurring around them, you know, that's what we listen to. That's what the goldfish listens to, the soundscape. And perceptually, I think they form a scene of all the sources and events within the soundscape. And this is auditory scene analysis. Now, they use this for orientation to tell them where they are, what predators or prey are nearby, and other things about their surroundings, such as its size, the presence of sound-reflecting features, etc. You know, if you can imagine walking into a room blindfolded and you don't know where you are, but someone hands you a basketball, and you bounce it on the floor once, boom, you immediately have all kinds of information about what the room size is, what the room is like, whether it's got carpet on the floor, whether there are lots of sound-absorbing things around there. You have an incredible understanding of the room based on that one sound, the one bounce of that ball. And so I think goldfish and all animals are using for their orientation. It places them. You know, it just tells them where, you know, the question is, where am I? Well, just listen, and you'll hear where you are. And so the answer to the question is, every sound source that they can hear is what they listen to, or what we all hear from our environment every minute, and what we all listen to. This is why we have hearing. This is why we, you know, have ears. This is why all vertebrate animals can hear and have ears. And it's because every incidental sound in the environment is meaningful and can be used for orientation. And so the way I say it is that the function of listening to every source is to learn as much as possible about the objects and events in your world. You know, there's no limit to how much you would want to learn about your world. You just learn as much as you possibly can. And why? So you can behave appropriately to all the events and objects in your world. That's how to survive. That's how we all survive. We behave appropriately to all the sources of stimulation. Some are sound, some are sound and light. Some are taste, sound, and light. Some are smell, sound, and light. Some are just smells. But there are sources. They're really out there in reality. And the better you are able to determine those sources, the more able you are to act appropriately with respect to those sources and to survive. And so I think for goldfish, it's the same. That's why they have ears. And so I think the reason that all animals have ears, it's the same. And the purpose of the auditory brain, which Joe mentioned, it's organized similarly for all vertebrates. The purpose of all this, the auditory brain in the ears, is sound source segregation and auditory scene analysis. That's what your ears and brain are for. I just want to parenthetically remark that De Chavigna in the year 2000 wrote a very interesting paper in which he called the auditory system of human beings a separation machine. That's fundamentally what it is. It separates, it segregates sound sources. And so I think that his analysis applies just as well to goldfish. I think this is the really hard problem. To segregate these sources the way our brains do it is a very, very difficult problem. Artificial intelligence has made great leaps and bounds, and every moment it's increased in its ability to decode speech. Automatic speech recognition. You know, you call up anybody on the phone now, and all you get is a computer automatically recognizing your speech in the background. It's wonderful. It really works. But it turns out that that's an easy problem. That's the easy, easy, easy problem. And as soon as you have two sources competing, or you say something with coughing in the middle, the computer goes haywire. It says, no, I didn't get that. Please repeat it. You know, if there's any noise in the background, the whole artificial intelligence thing fails. Or if there's two or more sources simultaneously, everything fails. It just doesn't work. And so the hard problem is the separation problem, the segregation problem. It's what our brains and fish brains and all brains have accomplished over hundreds of millions of years. It's the capacity for scene analysis. And that's why our brains have so many neurons in them. You know, you can look at the brain and you can say, well, why do you need 10 billion of these things, these cells? What do? If you think of the cells as just little robotic decision makers, it's true. What's it all for? But if you think of them instead as of solving this really hard problem of segregation, then you could understand why they have to be so complicated and was so inscrutable. And maybe, I don't know, we might never really understand how the brain does this task, which is self-evident to us that it does. It's just part of our consciousness that it's really performing this task. But we might never know how it takes place because it's such a complicated thing. And it's just like in the nature of brains to do this. That's what they're for making sense out of real environments, real sound sources, real objects. And so without auditory scene analysis or without the segregation, I would argue that hearing would be almost useless. No animal would have ever evolved an ear because it would only work when there's one sound source. And so this is something I've learned from the goldfish. Thank you.
Thank you. If we have any questions, we'll take them briefly at the two microphones.
I have a question, no matter how relevant it is. Since sounds are just sort of vibrations in the environment, and goldfish don't have ears, they're just sensing the vibrations some other way. Have you ever thought about trying these same tests on humans that are deaf, that might sense vibrations some other way? You probably wouldn't have to condition them with shocks.
Yeah. Well, I think people probably have done this kind of thing. But the idea is, fish really do have ears, and they do have an auditory system. And even though the ears act differently than human or mammal ears, but it sounds like that would be, what you're suggesting would be analogous to trying to communicate information to people who are hearing impaired or deaf through some other sense, like vibration on the skin, and people have done that. And I think there's been a lot of research where you have pads of multiple vibrators that you put on your skin, on your back or on your arm, and it's sort of like Braille. It's sort of automatic presentation of a Braille-like message. And people have done that, and it works reasonably well, and people can learn to acquire information that way. But there's something unusual about the human sense of hearing, or the sense of hearing in general in vertebrate animals, that there's no real substitute for it. You can maybe get a certain distance with vibration on the skin or tactile, or like Braille, but there's no real substitute. It's kind of mysterious how compelling and important the sense of hearing actually is.
Great. Well, thank you again, Dr. Fay.